IOP injection is an experimental medication that has been developed by Megapro for use in adults with iron-deficient anemia, or IDA, as well as a magnetic resonance imaging contrast agent. This module will begin with a brief explanation of how the unique structure of IOP injection gives it a novel mechanism of action in the treatment of IDA, and how this is expected to reduce the side effect profile compared to existing intravenous iron products. Next, we will summarize the non-clinical and clinical data that have been collected so far on IOP injection. Then, we will discuss how IOP injections are prepared and dosed. We will conclude with a description of the packing, storage, and stability of IOP. There are multiple intravenous IV iron products currently on the market that are administered in the form of iron carbohydrate complexes. In these products, a mineral iron core is surrounded by a carbohydrate coating to stabilize the complex and minimize the release of labile iron, which can induce oxidative stress and immunologic responses. The stability of an iron carbohydrate complex depends on the product's molecular weight. Smaller complexes, such as iron sucrose, are only semi-robust and produce relatively large amounts of labile iron, thus limiting their maximum tolerable dose. Larger complexes, such as iron dextran, have a higher structural integrity and can thus be delivered at higher doses. Carbohydrate coatings, such as sucrose and dextran, have been associated with severe anaphylactic and hypersensitivity reactions. INFED carries the boxed warning shown here. In addition, iron carbohydrate complexes have been reported to cause hypotension, peripheral edema, infections, and iron overload. The investigational product, IOP injection, is a superparamagnetic iron oxide nanoparticle. Its matrix structure and particle size function analogously to large iron carbohydrate complexes, which should allow for large doses of IOP injection to be administered safely in a single visit, without the generation of free, unbound iron. Unlike iron carbohydrate complexes, however, IOP is coated with a biocompatible methoxyl polyethyl glycol silane group. This non-carbohydrate MPEG silane coating is intended to reduce the rate of hypersensitivity reactions. Multiple non-clinical studies have been conducted to assess the safety of IOP injection. Single-dose toxicity studies found a no observed adverse effect level of 28 milligrams per kilogram in the rat and a maximum tolerated dose of 400 milligrams per kilogram in the dog. Cardiovascular safety studies in dogs showed no clinically significant changes with IOP injection up to 25 milligrams per kilogram. At 50 milligrams per kilogram, decreases in blood pressure of up to 60% below controls were noted, all of which resolved within 40 minutes of starting the infusion. IOP injection did not show any potential for QT interval prolongation. A respiratory study in rats showed short-lived increases in mean respiratory rate and minute volume with a high-dose IOP injection. A 29-day repeat dose study in dogs showed that a rapid IV bolus injection of 10 milligrams per kilogram over 2 to 3 minutes resulted in potential allergic reactions. However, no allergic reactions were observed when IOP injection was administered as a slow IV bolus over 6 to 15 minutes. All genotoxicity studies have indicated that IOP injection possesses no evidence of mutagenic activity. No effects were observed in a panel of local tolerance tests. No direct carcinogenicity or reproductive and developmental toxicity studies have been conducted to date. The efficacy of IOP injection for the treatment of IDA was evaluated in a rat model study. A single dose of IOP injection at 40 milligrams per kilogram was compared to one equivalent dose of the commercially available IV iron therapy, Ferraheme. After 3 days, hemoglobin levels increased by 93% for IOP injection-treated rats, compared to 65% with Ferraheme. The serum iron levels, which are an indicator of unregulated release of labile iron into the body, were increased twofold in Ferraheme-treated rats and did not recover after 23 days. In contrast, IOP injection-treated rats had transiently increased serum iron levels that nearly returned to baseline levels after 23 days. This study thus showed that IOP injection was more effective and safer than Ferraheme in a rat model of IDA. Systemic exposure following single doses of IOP injection in both rats and dogs increased in a slightly greater than dose-proportional manner. This effect appeared to decrease following repeated administration. The half-life ranged from 0.5 to 1.9 hours in rat studies and 0.7 to 1.7 hours in dog studies. Radio-labeled IOP injection was detectable for at least 150 days after administration, with the majority of label observed in the spleen, whole blood, bone marrow, and liver. The majority of the label was excreted via the feces by 28 days after administration. Very little label was identified in the brain at any time point, suggesting that IOP injection does not cross the blood-brain barrier. IOP injection use in humans has been tested in one Phase 1 study. Clinical study IOP-CT-001 evaluated the safety and efficacy of single, low doses of IOP injection in healthy subjects for use as a liver-specific MRI contrast agent. A total of 18 subjects received a single dose of IOP injection across 3 cohorts. 10 subjects across all 3 cohorts experienced at least one adverse event, with a total of 22 adverse events reported. All of these adverse events were Grade 1, non-serious events, and none required any action to be taken during the study. There were no serious adverse events and no dose-limiting toxicities were observed during the 14-day monitoring period. On closer examination of the adverse events, 6 subjects experienced a combined 10 events that were considered treatment-related. A list of these events is shown here. The most common treatment-related adverse event was increased transferrin saturation followed by dyspnea. Note that among the 6 subjects with treatment-related adverse events, 1 subject had 5 adverse events, whereas each of the other 5 subjects had only 1. IOP injection consists of superparamagnetic iron oxide nanoparticles conjugated with MPEG silane. The molecular formula is shown here. The overall hydrodynamic colloidal particle size is 45 to 85 nanometers in diameter. The average molecular weight of MPEG is 2,000 daltons. The appearance of IOP injection solution is clear with a dark brown or black coloration. The specific gravity ranges from 1.0 to 1.1 grams per milliliter. IOP injection is formulated as a sterile, aqueous colloidal solution that contains superparamagnetic iron oxide particles with 20 milligrams per milliliter of elemental iron, coated with MPEG silane, as well as 40 milligrams per milliliter of mannitol and 2 milligrams per milliliter of L-lactic acid. It does not contain any preservatives. The formulation is isotonic with an osmolality of 260 to 340 milliosmoles per liter and a pH range of 5.5 to 7.5. IOP injection is indicated for adults with iron-deficient anemia and as an MRI contrast agent. Its specifically designed matrix structure and MPEG silane coating are intended to allow for large doses of iron to be administered safely in a single visit, while also minimizing the risk of anaphylactic and hypersensitivity reactions seen with existing IV iron products. Non-clinical studies have demonstrated that IOP injection is generally safe, well tolerated, and efficacious for use in iron-deficient anemia. The first in-human study of single, low-dose injections observed no serious adverse events or dose-limiting toxicities. All adverse events were non-serious, grade 1 events. The most commonly observed treatment-related adverse events were increased transferrin saturation and dyspnea.